Hey Grandma, you know old corn Tenny? He seems very harsh, lean and mean.
Yes, I know Mr. Teosinte. He is quite a nice corn, but has a hard, tough skin, which is why a lot of people don't like talking to him. Did you know that he is actually related to us?
Really?
Yes. Long, long ago, his great ancestors were our ancestors. I'll tell you more if you come sit down.
Okay, so if we are related, why do we look so different?
Well, there are many possible reasons. Our family got picked to settle here, while Mr. Teosinte continued to sail the world. This meant that he grew up in different environments to our species. Something so small as an increase in the fibre of our diet could have contributed. We know that we look different, but only recently the actual differences between us were revealed. Some very smart scientists came by and found that we have small variations in our DNA.
Different DNA?
Over time, our DNA has changed and evolved, adapting to the environment. Our evolutions have resulted in tastier and easier to produce corn. This means our species, the maize-ing, experienced domestication from the progenitor Teosinte. Now why don't you ask your grandfather about how the scientists found differences between our species, the maize-ing and Teosintes? After all, he worked with them.
Thanks, Grandma. Bye! Hey Grandpa, Grandma says that we're very similar to old corn Tenny, but we look so different. She said that you worked with the scientists last holidays.
Whoa, slow down there. How about we go through this from the top, eh? Alright, so Grandma explained what our DNA does, right?
Yeah, a bit. Our DNA can change over time and helps us taste good.
Yes, that's true. It is like the building instructions of our bodies. DNA must tell different parts of you to be made in different ways. To do this, it expresses genes differently in our systems. Our kernels here are built from instructions given by many genes, one of which is TGA1. This gene is expressed in our kernels, but not in our stalks or stems.
I don't understand that. How do we express our genes differently?
Let me draw you something here. This purple rectangle represents a piece of DNA. The part in orange is one of our genes. DNA has regions called promoters that are upstream of genes. Promoters can influence gene regulation by assisting in translation. The effectiveness of these promoters is just one way a cell can control gene expression.
So that's how we can control our genes.
Yes, that's one factor. DNA can also undergo mutations. If the promoter is mutated, its properties have probably changed, so its function will also change. Thus, the expression of downstream genes might be different. This can help generate genetic diversity, which is very important for a population.
But what if the mutation is inside one of our genes?
Ah, yes. Well, these mutations can affect the job that a protein goes to do when it is translated. If a protein is changed, it might have different properties like being acidic or basic, and the stability of the protein could change. Some mutations can be deleterious and cancer-causing. Negative mutations that affect genes expressed widely in an organism can be incredibly dangerous. However, mutations that occur in genes affecting a localized area of an organism won't be as severe. The scientists that came over were from a very special researcher's lab, and his name was George W. Beadle. Mr. Beadle was the first person to really challenge the idea that our ancestors were unrelated to the Teosintes. Before him, most scientists thought just like you did. We had too many significant structural differences to be related. Tenny has hard copulate fruit cases, while we have soft kernels on our surface. His different DNA tells him to build hard silica glooms, which don't break down when passing through animals' digestive tracts, giving him that hard, stern look you see. But we have similar kinds of hardness.
Wait, really?
Our cores are made up of the same hard organs, but with slightly less silica. The scientists first asked for corn samples. 12 DNA samples from Tenny's family, the Teosintes, and 16 from ours, the maize-ing. We used this to make a map of our genomes.
Why do they need a map? They're not going to get lost.
No, not a map like that. One strand of DNA might have been broken up, so by combining multiple DNA pieces we can rebuild larger, more complete DNA contigs, eventually getting enough information to call it the genome. When the DNA constructs from the Teosintes and maize-ing were aligned, 7 base pairs were found to be different.
So that means 7 mutations, right, Grandpa?
That's right. 1 mutation was found in our TGA1 gene, while 6 single nucleotide polymorphisms were identified in the promoter region. The mutation within the gene was a substitution of a basic lysine amino acid to an uncharged polar amino acid, asparagine. The mutations in the promoter might change how the gene is expressed, but from previous research into the domestication of tomatoes and cauliflower, the mutation within the TGA1 gene was expected to have the largest effect on the structural changes we see today. Research into the effects of singular genes on the domestication of other plant species has shown to influence the evolution of certain traits in these species. For tomatoes, the gene FW2.2 has been identified to control fruit weight, and the buccal gene in broccoli can define the inflorescence structure. The scientists left Zeville with the stronger idea that 7 nucleotide polymorphisms, or even just one base pair substitution, can have a large effect on the phenotypic changes leading to the domestication of us, the a-MAIZE species. Securing a reliable source of food is important when supporting a large population, so I hope you see that this research was very important for food security into the future. Now I'm starved. It must be time for dinner.
Oh no, not more beans and chia seeds. Too much fibre. I'm sweet enough already.